He's working as a research scientist in Organica Biotech Private Limited, Mumbai. He has done his PhD and in the specialization in environmental bioremediation from Vellore Institute of Technology, has published 7 research papers, 2 review articles, and 1 book chapter. He has been awarded the National Biotech Youth Award in 2023 by Microbiology Society India for research contributions in the field of microbiology. He has been awarded the Young Scientist Award, Young Research Award, Raman Research Award, and Innovative Research Awards throughout his research career. So the platform is now all yours, Dr. Bob. So you can please begin. Welcome you once again on behalf of MBSI.
Thank you, ma'am, for the warm welcome. So, a very good morning to one and all present here. I am Dr. Mrinal, a research scientist from Organica Biotech Private Limited. So, as the part of this PhD series, today I got a privilege to share with you all the ideology that I have proposed during my PhD and I have worked. So, I'll share my slides and then we can carry forward with the presentation. Is my presentation visible?
Yes, yes. You can make full screen. I guess.
Yes. Yeah. Now it's okay, right?
Yes.
Yeah. So today I'll be presenting my PhD work on the series titled BioBlitz, a teamwork for toxic trio cleanup. So today's roadmap of the presentation will start with a small introduction on how the environmental is been into a threat and what are the different perspectives. Then it will be a hypothesis.
May I just interrupt you for a minute because, like, your awarding year was not mentioned in your writing, so I could not say to the audience. Please, if you could mention your PhD award year so that, like, people know that these recent things like that. You follow
Yeah,
me?
sure.
Yeah, just
Sure.
mention the year. Yeah.
Sure. So my PhD basically was on the environmental bioremediation under the guidance of my research supervisor, Dr. Saravanan S, Associate Professor, VIT Vellore, and my co-supervisor, Dr. William Jabez Osborne, VIT Vellore. I have been awarded with my PhD degree in 2023. Is it okay, ma'am?
Yes, sure. Yes, sure. Yeah. Thank you. Yeah.
Yeah, sure. Thank you. So today's presentation the roadmap of today's presentation will start with the introduction, followed by a hypothesis that was the major aspect of our work, followed by the objectives, then the discussion on the results and the conclusion. So a small introduction about what exactly environmental remediation is, what are the different pros and cons of environmental. So as we all are aware about the major sources of heavy metals, which was the main target of my PhD into the environment, is either through natural, agricultural, industrial, domestic, and miscellaneous sources. Here, specifically, apart from natural, the agriculture and industrial are the major sources through which the entrance of toxic heavy metals into the environment has been found more. So how does it comes into this story? So it comes through direct pollution, that is through soil contamination, and indirectly either through water or air contamination. So when these heavy metals have been discharged, they directly have agroecological consequences on the soil health as well as the plant performance. So how it impacts the plants and soil, we specifically see. So in soil, it reduces the soil fertility, it modify the microbial population and processes, modification of carbon mineralization, and several transformation into nitrogen and other essential sources. When it comes to plant, it induces the oxidative damage, anomalous genetic behavior, modifications of signaling and biosynthesis pathways, and abnormal response, reproductive response. So the reduced crop, this like representation which I have shared is from Srivastav et al. 2017. Another major aspect when it comes about the deposition of these toxic metals into the soil is basically found majorly due to uh, yeah. So when it comes to heavy metal toxicity, the major sources of deposition of these toxic metals into soil is from into different variations of the regions. Like it will be either domestic or when it comes to mining, agriculture, wastewater industry. Next was the majorly what are the different sources of these toxic metals into water. So heavy metals and metalloids, 32% of deposition as per a report by Down to Earth was been given specifically into the regions of South India, which was our major target of the study. So as I have told, it is affecting the soil as well as water and other ecological factors. It is somewhere also having a major impact on humans because directly or indirectly these toxic metals are in coming contact with humans. So when it comes into specifically into males, it is causing vomiting, diarrhea, intravascular hemolysis, reduced thyroid hormones, lung cancer, cardiac arrest, bone decaying, renal dysfunction, and erectile functions. When it comes to specifically females, it is severely causing the issues into menstrual disorder, female infertility, um, then spontaneous abortions, endometrial cancer, and breast cancer. In small infants, specifically at their initial stage of the thing, it is causing vomiting, diarrhea, blue or black lines in gums, dehydrations, chills, low body temperature, stomach pain. So, as the major role in my study of the heavy metals was that we have targeted three metals, that is lead, nickel, and cadmium. So, these are few of the real-time figures we can see, which is caused specifically due to lead exposure into different regions of India as well as specifically global. Main reason of choosing lead, nickel, and cadmium was specifically lead and nickel was that in many parts of South India, from 2017 till 2020, it was been reported that each category is been suffering from severe effects due to toxicity, which was been confirmed that different symptoms through which they were going, and when the medical tests were being performed, the concentrations of lead, mercury, cadmium were found into the body of the patients who were coming. Similarly, several physical symptoms were also been visible. So these are some of the real time reports that we have came across. So based on the literature, whatever we have done, our next target was like what are the creatures that are basically been getting affected into the environment, specifically soil or water. So we all know that earthworms are being reported as the ecological engineers. So basically, what they are doing is they don't need any specific kind of food consumptions and all. They all totally depend on the matter present into the soil. So somewhere it was been observed that these earthworms are also been going through a lot of hazardous effects due to the toxic heavy metals as one of the part. Similarly, due to excessive stress of these heavy metals into the soil, the earthworms have somewhere adapted themselves into such a way that their growth physiology have somewhere been known to have the several enzymes or specific um, probes that are helping to clean up such toxic materials. A report, Times of India news report of 2022 have shown that agricultural soil in Karnataka over 5 years have been increased the population of earthworm, which basically is helping in good fertility of the soil. The next second report basically was by Tanzania 2018 and the Economic Times of 2019, which clearly shows that heavy metals in vegetables. So basically, what is happening that, as I have told, that these toxic heavy metals are being deposited into the soil from different sources, and thus in many of the areas all over India, the cultivation of different vegetables is into the either nearby the water bodies, or somewhere the water that has been used throughout the process is being either contaminated. Thus the maximum vegetables like brinjal, tomatoes, cabbage, which has been regularly consumed by humans, have been reported with excessive amount of the toxic metals, under which our three targeted metals were also been observed. Next was when it comes to water, as water bodies are mostly contaminated. So it was been observed that the sudden death of the fishes is just not because of some kind of physiological or any kind of diseases that has been coming, but excessive contaminated water, which specifically was also been due to the concentration of severe metals like cadmium, lead, um, chromium and all, is been somewhere affecting these water bodies also. So next was the target area of our study, that was Ranipet. So Ranipet comes under Tamil Nadu, in the state of Tamil Nadu, which is known as one of the most polluted area due to the tannery locality as well as other industries nearby. Specifically, the agriculture area, which is the common source of earning of the local people in Vellore as well as Ranipet, is nearby the agricultural locality. Thus, day-to-day water consumption for their personal as well as their agricultural aspect is the same source. Thus, we also come across that the mm, uh, toxic contaminants with the higher permissible limit is being found into a higher amount in this region. Thus, our sample location and our experimental location was also from Ranipet, Tamil Nadu. So, based on the literature that we came across on the metals, or what are the different creatures that has been every day affecting, we have come up with a research study which specifically explains that the emission of our targeted metal, that is lead, nickel and cadmium, is through different sources, but specifically the industries. Thus, these emission of lead, nickel, and cadmium through inhalation goes into the air, through discharge into the soil, it is coming in contact, and also it is running off into the water. Thus, the inhalation, discharge, and runoff is somewhere directly or indirectly coming in contact with humans through consumption. Thus, overcoming this issue, the second thing is also the plants, which are the majorly growth factors when it comes to when it comes to water or soil. There are several like mechanisms through which such toxic metals directly enters into the plants, and somewhere, as I have told, that several vegetables are also been reported to have the concentrations of heavy metals, they are being consumed by humans. Thus it is a kind of chain through which these metals directly, indirectly, through consumption or anything, is affecting humans, as well as somewhere it has destroyed the peace of the nature. So what can be a probable source or a solution for this? Thus, a heavy metal resistant plant growth promoting bacteria in combination with different study models like earthworms, plants, fish, was the major aspect of our study, which will somewhere increase the fertility of the soil. Also, it will help it to improve the plant growth, which is another aspect of the study, as plants were also been affected throughout the study, which was been observed. Next was the product formulation, which is an essential factor, and the pest control, because it is also one of the aspect which is directly or indirectly harming the plants. So, based on the research hypothesis, we have come up with some objectives throughout my study. That was the isolation and screening of a lead, nickel, and cadmium resisting biofilm forming plant growth promoting bacteria from the contaminated sites. Next was the toxicity assessment of lead, nickel, and cadmium on the seeds mentioned. Four different seeds we have selected, and zebrafish model augmented with potent bacterial strain VIT-MSG3. Next was the examination of toxic effects of lead, nickel, and cadmium using earthworm model. After that, we have focused on assessment of lead, nickel, and cadmium using phyto and rhizoremediation associated defense mechanisms. After that, we have also tried to focus on a paradigm of lead, nickel, and cadmium uptake using the triple biosystem of C. zizanioid, Eisenia fetida, and Bacillus zeaminensis. After that, we have focused on the soil bacterial community using metagenomic analysis. And the last basic study that we performed was on enhanced efficacy of Bacillus zeaminensis upon augmentation against specific, Spodoptera litura and toxicity reduction by its oxidative enzymes. So before I go into discussing each and every study reports that we come up, I would like to clarify few points here itself, so it should not create a confusion in the upcoming slides. So lead, nickel, and cadmium were my targeted metals throughout my PhD study. When it comes to aqua, what we have toxicity study we have performed. So we have focused on these four for the germination assay. We have selected these four seeds. After that, Danio Rerio was or zebrafish was been used for the toxicity study on in water. Earthworm model, when we performed the study, we used Eisenia fetida as the model. When it was plant study, that is phyto and rhizoremediation study, we have used Chrysopogon zizanioid, that is vetiver, and Bacillus zeaminensis was the potent isolate that we procured, and Spodoptera litura is the larvae on which we have uh, tried to check the efficiency of our potent bacteria into with the perspective of agriculture. So moving towards the like objective one, which was basically the major like part of the study. So as I have told that that Ranipet area was one of the most polluted area. Apart from that, the sample location for isolating the potent bacteria, we have selected five different places. That was paddy field from Katpadi, agricultural field of Ambur, Tamil Nadu, textile industry Erode, Tamil Nadu, leather industry Varthur, Karnataka, and Pulinthangal Lake. So the concentrations mentioned for lead, nickel, and cadmium were been analyzed using the samples what we got from there, and we have performed the analysis, AS analysis. Figure 1 and figure 2 shows the different sample collection sites and the site locations, as well as the randomized grid technique, which is preferred for the sample collections. So moving towards the next slide, that was the morphologically distinct colonies on MSM plates was been obtained, followed by the load count was also shown, and these are the 10 heavy metal resistant, that is lead, nickel, and cadmium resistant strains isolates what we have observed. So the after that, we have performed a lot of screenings which have been visible into the table 3. That is, we have performed maximum tolerable concentrations, biofilm formation, quorum sensing, plant growth promoting a test like ammonia production, phosphate solubilization, IAA production, hydrogen cyanide production, siderophore production, ACC deaminase, and lead, nickel, and cadmium specific gene amplification. So we have performed all these experimentations for all the 10 cultures, and the profile which I am showing here into the table is for the potent bacteria that we have used throughout our study, because VIT-MSG3, that was the culture 3 what we obtained from Pulinthangal Lake, have shown the maximum concentrations of metal that we have tested was 4000 mg per liter of lead, nickel, and 500 of cadmium, followed by biofilm. It was able to form a strong biofilm. Quorum sensing, it showed large and short chain molecules positive, All the PGPR tests were been found to be positive in the presence of different concentrations of metals. Similarly, the lead, nickel, and cadmium specific genes were also been found to be amplified at 500 base pair, 1058 base pair, and 1141 base pair. Now, I would like to highlight a major thing before going to the next slide, that the all screening experiment were being performed here, either it is PGPR or biofilm, everywhere, every specific experiment was being performed in the presence of the targeted metals which were being used into the study, because it is an essential aspect when it comes to we are working with any contaminant, it should be present in all the experiment. So into the next slide, I would like to show the some of the figures that have shown a very positive response in the study. So this is a qualitative assay and quantitative assay of the biofilm with different concentrations of metals that we have used. So figure fifth show the biofilm formed by VIT-MSG3 using test tube assay in different concentrations of metal, that is lead, nickel, and cadmium. Similarly, figure 6 shows the biofilm assay of into the microtiter plate that was been quantified to check the soluble biofilm efficiency. Next was the in situ biofilm visualization. That is also an essential factor, because through test tube we can see a band over a tube which determine its efficiency to produce a biofilm. Through solubility, we can come to know the amount of soluble biofilm that has been got, and in situ determines the actual visible biofilm. So we have performed it through light microscopy, scanning electron microscopy, and fluorescence microscopy. So the left side image in ABC shows the control, and right side image shows the VIT-MSG3 potent bacterial film. Next image 7 shows the quorum sensing molecules, because when it comes biofilm, when we talk about biofilm, quorum sensing works hand into hand, because when there is cell to cell communication, then the efficiency of forming a biofilm is higher. So as I have said, the bacteria was able to form a quorum sensing large as well as small molecules both. So the purple color change shows confirms the presence of CVO2 CVO26 against CVO26, and when it comes to the green color, it shows against NTL4, that is the long chain forming control. Next was the amplification of gene, as I have told. So VIT-MSG3 was successfully able to show the amplified genes, and certain enzymes were also been checked. Further, just to confirm whether what was exactly the strain, so we performed 16S rRNA gene sequencing, and our strain VIT-MSG3 was able to was found to be similar with Bacillus zeaminensis, which is a gram-positive rod-shaped motile bacteria. So these are the few results of my objective one which I have showed, because it might have made the data so clumsy. So whichever I felt was essential, I have showed. So moving towards the summary of my objective one, it clearly states that isolation of lead, nickel, and cadmium resistant PGP rhizobacteria was performed. And among all the 10 strains, VIT-MSG3 was selected as a potent strain on the basis of several molecular as well as phenotypic analysis. VIT-MSG3 was the excellent tolerant of lead, nickel, and cadmium till higher concentrations, and also a short chain and long chain bacterial biofilm forming ability. It was screened out as a strong PGPR due to its ability to show the activity for qualitative as well as quantitative assays in the presence of lead, nickel, and cadmium as well as genes. Further, upon gene sequencing, the strain was identified as Bacillus zeaminensis. Now, through an objective one, we came up with a potent strain which was able to work and boost up these toxic metals. So so the next step of the R study was to check its toxicity on the fish model as well as on the different seeds. So we have specifically focused 4 different seeds based on the literature as well as the previous studies that have been performed in our laboratory. And figure 11 specifically shows the setup of different seed germination assays with different treatments, starting from a normal water control, then controlled broth, different concentrations of the bacteria, supernatant and pellet, then in the presence of heavy metal with bacteria and without bacteria. And it from our study, uh, con- we concluded that the supplementation of VAT MSG3, either it is in the form of cells or supernatant, and even into the presence of different stress condition, it was able to grow and gave a good germination rate with an increased relative plumule and radical growth. So thus, through seed germination, we concluded that yes, the efficiency of the bacteria in germination as well as to come up with the growth parameters which are essential for the plant have been cleared. The next strategy was to go for a zebra fish, that is a fish study model. So here is the work plan for the study which I have used. Basically, we kept the fish for 15 days adaptation into the light is to dark cycle, because it is an essential factor when we transfer some creatures from their natural living system into artificial. So 15 days, after 15 days adaptation, we have basically different setups, four different setups. One was a control which was a normal water with fish. Then setup one was different concentrations of the heavy metal, that is lead, nickel, and cadmium of 50, 100, and 150 mg per liter, and we exposed the fish individually. Next setup was only bacterial treated water with bacteria, and fish is exposed. And the setup two was with different concentration of targeted metals with bacteria. The study was been performed basically for 20 days. And at each interval of 5th, 10, 15 day, we have examined several parameters like physiological changes, histopathological changes, DNA damage through comet assay, SEM analysis, bioaccumulation factor, uptake of these metals, enzymatic assays, and physicochemical water analysis. So here into this presentation, I would like to show some pictures that will depict the reports or studies what I have been concluded here. So here, figure 12 shows the experimental fish what we have performed. So the yellow and red color arrow determine the severe breakages and damages, visible damages that were being observed into the heavy metal treated exposed fish. Specifically, the mortality of these metal stress under metal stress exposed fish was found to be so less that till 10th day the fishes were found to be dead. The control as well as figure A determines the control fish which was found to be fully active. Similarly, E, F, G, and H shows the bacterial treated in presence of heavy metals fishes, which clearly have been found to be like alive till the 20-day study and beyond it, with no kind with no any kind of visible damages or anything. Further, to confirm whether there is any kind of deposition of these metals which we are talking about into the gills or somewhere, so we have dissected the fish. We have taken the gills and we have performed scanning electron microscopy. So figure A and B under scanning electron microscopy shows the gill of control, which was found to be undisrupted and clear, as IDAX confirmed there was no any deposition. C and D shows the gill of bacterial treated uh, bacterial treated of bacterial treated fish, which have shown the slight deposition of lead. Interestingly, there was no kind of disruption into it. E and F determine the heavy metal treated fish gills, which clearly shows a severe deposition onto the gills. And when we focused on one of the part and confirmed IDAX, we came across that lead concentration was found to be higher, followed by cadmium and nickel. So, apart from that, as I have talked about histopathological or antioxidants and all, so when these experiments were being performed, so we came across that seed germination assay visualized the enhanced growth of all the four seeds. As I have already told that the seed germination confirmed us that the plumule and radical growth was found to be increased with the increase in germination rate. Similarly, the poor growth into germination factor was been found when the seeds were being treated with lead, nickel, and cadmium, which have a greater impact. On the other hand, when we performed a fish study, the physiological parameters have shown a decrease in length, weight into the heavy metal treated fish, and also the mortality was found to be obtained earlier. But upon supplementation of VAT-MSG3, it was found to be somewhere stable. Scanning electron microscopy, I have shared the figures, so it was been clearly observed when we perform DNA damage through comet to check whether there is actually any kind of uh, the confirmations through the study. We have come across that there was the head and tail damage elongation was found to be higher when the fishes were exposed with only heavy metals, and histopathological analysis also have shown a severe disruption. Sorry, I am not able to share the photos of histopathological because it was a huge data and it might have been created a slight confusion. Overall, the toxicity study in this phase of our study confirmed that yes, the bacteria somewhere is helping to reduce the toxicity, as the oxidative stress through enzymatic tests that we have performed like SOD, POD, APX, GST was found to be minimal. Next step of the study was to check whether the uptake of these toxic metals from the soil can be accumulated into the earthworms, so that we can somehow detoxify the soil from these heavy metal, as well as somewhere it will help to biotransform these metals into less toxic or non-toxic forms by the earthworm. So earthworm was being used Icenia fetida. They were been kept into adaptation study for 15 days, followed by three different setups with different concentration of metal and bacteria were been exposed. The earthworms were been exposed. The study was performed for 28 days, followed by different parameters that we have checked. So, examination of visible damages into the earthworm was been checked, and you can see that in figure A, first figure 1 is of control, which is showing a good visible growth of the earthworms, followed by 2, 3, 4 have shown the 2, 3, 4 have shown the earthworms with bacteria and heavy metals, and 5, 6, 7 have shown the only heavy metal treated earthworms, which clearly showed the breakages as well as severe damages into the earthworm. Next was the scanning electron microscopy of midgut. So here A shows the control earthworm midgut. There was no deposition. Followed by B, also have we have shown the zoomed or the magnified part of the earthworm control, which have showed again there was no kind of any deposition on the gut. C and D figure shows the heavy metal, that is lead, nickel, and cadmium treated earthworm gut, which clearly show a disruption. And figure D show some kind of deposition on the surface of the gut, which was been confirmed, and it showed that heavy metals like lead and nickel was found to be higher on the section that we have majorly magnified. Figure F and G shows the heavy metal and V8 bacterial treated earthworm gut, which clearly show the rod shaped deposited bacteria on the gut of the earthworm. Similarly, after that, we have also at each interval of 57 day, 14 day, 28 day, we have taken the earthworm samples from each setup trace. We have went for acid digestion of those earthworms as well as the soil to check the uptake of heavy metals into the earthworms, and we have performed atomic absorption spectroscopy, that is AAS analysis. So we came across that till 28th day, there was almost 30 to 40% of increase into the uptake of heavy metals into the earthworms from the soil when it was been exposed or supplemented with VAT-MSG3 bacteria, which clearly shows that the bacteria somewhere is working on the mechanism of biotransformation, where the toxic metals and toxic metals and bacterial supplemented soil, when it's been consumed or when it has been coming in contact with earthworms, the toxic metals that are being biotransformed into less toxic or non-toxic form are being accumulated into the earthworms, which is somewhere neither harming the earthworms as well as the maximum cleaning and boosting of these toxic metal has been happening. The gut tissues were also been found to be undisturbed when the histopathological analysis was been formed, overall, when it comes to oxidative stress, the antioxidant enzymes were also been performed, which clearly gave an idea that the upregulation of these activated enzymes is somewhere reducing the oxidative stress caused due to the heavy metals. Now, the earthworms somewhere gave us the promising result, and at this third step of the study, that the uptake of these heavy metals can be accumulated into the earthworms without harming the earthworms too. So our next step was, can there be any source of plants that would be helpful in the uptake of these toxic metals from the soil also? So next step of our study was to go with phytorhizo study using a plant. So in this study, the plant which I have focused was Chrysopogon zizanioid, which is also known as vetiver or khas. So the rationale behind selecting this plant is like, when we talk about any of the study, the final output which should come have some value. So the khus basically, or vetiver, is somewhere a fragrance creating plant. Thus, after the study, we can somewhere use these treated plants also as a value added product. So here in our study, what exactly we did was, like, we have performed a study for 45 days with different concentrations of metals in presence of bacteria and without bacteria. And at each interval, we have collected the samples, plant samples, and we have performed different series of experiments. So physiological alteration in plants clearly showed that the control and bacterial treated plants have shown the similar root and shoot growth, which was found to be excellent with higher chlorophyll content, and relative water content was found to be also higher. When it comes to only metal treated plants, it was found to show a very poor growth with less chlorophyll content, and also the plants were found to show the poor growth of length into root and shoot also. uh, we have also performed several different assays to confirm the plant, and the uptake study visualization was also been performed through scanning electron microscopy of root and shoot. So ABCD shows the root and shoot of control plant, which clearly shows that there was no kind of excessive plant like deposition of the metal. EFGH shows the phytoremediation plant, which have shown the less uptake of the metals into the root and shoot, which was specifically found of cadmium only and nickel. Interestingly, IJKL figures show the same index of root and shoot of rhizoremediation study, which shows clear deposition of the heavy metals of lead, nickel, and cadmium into root and shoot. Thus, it confirmed to be a root as well as shoot accumulator. Further, similar pattern which was been followed for earthworm, we have performed here for plants also. We have performed antioxidant assays to check the oxidative mechanism into the plants, which clearly shows that there was a positive correlation of SOD, POD, CAT, APX, GST. These are the enzymes which have shown an increase till the 45th and 60th day, which have somewhere hypothesized that the oxidative stress was found to be reduced due to the presence of uh, bacteria. Similarly, there was an 80 to 90% of uptake of lead, nickel, and cadmium from the soil into the roots and shoots of the plant, which have given a positive impact of the study, that the plant what we have targeted in our study have the ability to overcome, or to show the higher uptake of these toxic metal from the soil into the plants. So the next step was that if we are getting a 70 to 80% uptake using earthworms detoxifying the soil, we are getting 80 to 90% of uptake into the plants from the soil. What if we club earthworms, plants, and bacteria together for going, which will help to give to translocate and bioaccumulate maximum toxic metals from the soil, and it will clear the soil. So in this study, what exactly we had into our mind was like, we have performed this study in combination with earthworms, plants together, and bacteria. So figure 17 shows the 60-day plant of the study, which clearly shows the control and different treatments of the bacterial treated plants have the similar root and shoot growth here. Sorry. Then EFG shows the vetiver plant of 60-day, which clearly show the poor growth of root, as well as shoots are found to be dry, which was been confirmed after chlorophyll content, that the content was found to be very less. So it might be a probable chance that the excessive concentrations of metal that we have exposed into the study is somewhere impacting the physiology of the plants. Next, figure 18 shows the same setup exposed earthworms, where control and bacterial treated earthworms have shown the similar growth pattern, and the poor growth was found to be into VIT-MSG3, that is bacterial non-treated earthworms. So, in this study, as I have told that we have performed a combined trio study, here we came across with a very amazing result was when earthworms and plants in together combination with bacteria were being used for detoxification of the heavy metals uptake from the soil. 90 to 95% of removal of lead, nickel, and cadmium was been obtained here, which was somewhere a positive correlation that showed a clear image that the earthworms as well as the plants were found to be alive with no severe damages or disruptions or any kind of stress related disorders into them, which showed that yes, somewhere bacteria is helping to nurture the growth of plants and earthworms, and it is also biotransforming the toxic metals into less toxic forms of its, which is somewhere acting as a nutritional source as well as a non-toxic source for all the living biosystems used into the study. The next was the microflora. Also, we have to confirm whether, see, we are talking into the study that we have supplemented bacteria. But really, this bacteria is only the probable reason which is working. To confirm that, we have also performed the microflora study, where we have collected the rhizospheric region soil of the experimental plants and also the soil overall on the earthworm body. We have performed serial dilution, and we have performed checked the morphology of the bacterial culture obtained on the spread plate, and a single the bacteria colony which was found to be more was have shown the similar traits with that of our strain. Similarly, the DNA damage in earthworm was also been found when it was also been nullified with association of the bacteria, which confirmed that yes, somewhere the bacteria in combination with earthworm and plant is somewhere helping. So this study led to the development of a better in situ remediation strategy and a better understanding of interaction between earthworm, plant, and bacteria for enhanced remediation. So till this step of the study, we confirmed that somewhere our targeted metals were found to be removed at each step with a increased uptake potential and with less effects on the biosystem that we have used. Thus, the next part was to validate and confirm that the maximum microbial diversity throughout the study, what we have performed in presence of the bacteria, should confirm the bacterial classification that should be somewhere showing a similarity index with our bacterial classification. Next, thus, based on that, we have performed a systematic study of alteration in soil bacterial community using metagenomic analysis. So what exactly in this study we have performed? So what exactly was the strategy into the study was that after the association study for 60 day of lead, nickel, and cadmium bacteria, earthworm, and plant, we have collected the soil samples from control, the only metal treated soil sample, and the soil sample from the bacteria and metal treated setup, and we have uh, analyze the metagenomic analysis. So basically what metagenomic analysis explain is, so first step into metagenomic analysis is for a preparation of library from the extracted DNA, that comes up with which talk about the sharing of the dsDNA, then amplification using V3 to V4 region using specific primers, followed by addition of adapter sequences at I5 and I7 thing. Next step is to go for cluster generation. So it is something like It is composed of oligos, where DNA fragments are hybridized and copied into a flow cells coated with oligonucleotides, which forms a clonal cluster. Next step is sequencing. So what exactly we do is like Illumina labeled sequence, what we performed here, followed by the last step is data analysis that basically confirm the functional profiling of the study. So here, experimental samples. So this is these are the tables specifically show the physiological analysis of experimental soil. So table 7.2 shows the library profile of all the experimental sample. So it specifically show the base pair region that was been targeted into V3 to V4 region, the average size of the base pair, and the total operational taxonomic units that have been observed. Then table 7.3 shows the specific level of alpha diversity, followed by table 7.4 shows the physiological analysis of experimental soil, which clearly shows that lead, nickel, and cadmium concentration was found to be reduced into the rhizoremediation soil. So next is the abundance level in control samples. So what exactly abundance level determine? So as I have told that we performed the a genomic analysis of the soil sample. So we come up with the phylum, that is all the phylum lists that has been performed into our samples. So here the control sample of 0th day and control sample of 60th day have clearly shown that the maximum abundance of the prominent abundance of the phylum was of proteobacteria with 25.5 percentage into the on the 0th day, and on the 60th day the proteobacteria was found to be standing there at the first position with an increased 5%, that is 30.7% of abundance. Next was the absolute abundance of the prominent phyla into the phytowormy remediation setup. So on the 30th day, the prominent phyla was found to be proteobacteria with 36% abundance. And the 60th day, it was again the proteobacteria with slight 1% of reduction into its abundance. Interestingly, the study took a turn here because the major uh, target of our study was what microbial or what is the microbial diversity or bacterial diversity that we get into our bacteria treated soil samples. So figure A basically here shows the rhizovermidation soil sample of the 30th day that was been given for the analysis, and it showed that proteobacteria was found to be 44.59%. So somewhere the phyla proteobacteria was found to be prominent till the 30th day. Interestingly, on the 60th day, the firmicute have overcome the proteobacteria with 56.55%. Now, what is the correlation of firmicutes with our bacteria? So, moving, so giving an idea about it, that bacteria Bacillus zeaminensis comes under the phyla Firmicutes. Thus, somewhere the probable reason of the confirmatory aspect of the study was that the Firmicutes to overcome proteobacteria at the 60th day confirms that somewhere our potent bacteria is the prominent strain which is helping in detoxification of the soil under lead, nickel, and cadmium stress. So comparison of phyla between the samples was been performed, and it was clearly observed that till 60th day of rhizowormy remediation, the firmicute was found to be shown the higher abundance. So based on that, the firmicutes, if we see, the phylum firmicute shows the class Bacilli, order was Bacillus, family is Bacillicea, and genus is Bacillus, and species specifically is Bacillus firmus group. So our bacteria Bacillus zeaminensis come under the taxonomic classification of the firmicutes with the similar pattern which has been shown into table 7, which confirm that yes, somewhere Bacillus zeaminensis VAT-MSG3 supplementation was have the positive impact on the detoxification of the heavy metals. So next was the heat map, which have shown the operational taxonomic units of the samples. So here red color shows the less red color shows the less abundance OTUs obtained, and green color shows the higher abundance OTUs obtained. So here firmicutes with 56 was found to be higher, and reduced was chlorobene. So summary of the objective was that somewhere this study confirmed that proteobacteria was found to be prominent till 30th day into rhizoviremediation. But interestingly, on the 60th day, its alterations by formicutes have confirmed that our bacteria was found to be potential. Thus, the whole study was validate that whatever we have performed with bacteria or with only heavy metal, with only earthworms, or somewhere we have only performed with plants and the combination of earthworm, plants, and bacteria. Thus, it validated that yes, the bacteria has the potential to club up with all these biosystems and detoxify the metals. So, the next and the last objective of my study was a kind of slightly different work, because as I have said that the major sources of heavy metal deposited into the ecosystem targets the environment and agriculture also. So somewhere when it comes to soil, water, it is also a major source when it comes to agriculture. So during my studies, when I was been performing at my institute, I came across that into the greenhouse, into our agricultural department was facing a issue of a larvae that was Podoptera litura infestation on many of the plants there. So, with an discussion, with a discussion with the entomologist, Dr. Lindsay, who was one of the advisor for this study, she like we have come up with a plan. Can this resistant bacteria have the efficiency to treat this larvae also? So, just with an hypothesis into a mind to do something in for contributing into the agricultural also, we have designed a small study where we have performed a leaf emission assay into the lab, where castor leaf was been like taken as a feed, which was been treated with Bacillus zeaminensis, that was my potent bacteria. Next was Bacillus thuringiensis, that is the positive control, and a common pesticide that has been used overall into Tamil Nadu was been used. I would like to give an insight on the study that specifically this specific larva Spodoptera litura was found to be in many farms of Vellore district. So farmers were facing issues that was also one of the reason to perform this study. And when we performed this study where we have exposed the larvae on the bacteria and different treated plants like leaves into the plates, as you can see. So the physiological analysis clearly showed the control larvae was found to be somewhere active, and BT treated also larvae have shown the good proper like have shown the proper growth, and the treated bacterial treated and pesticide treated pesticide treated larvae were found to be slightly like the severe mortality and death, because the red arrows clearly show the damages. So here the scanning electron microscopy of the midgut was also been performed. So for A and B, there was no deposition into the midgut of larvae. Uh, then C and D basically shows the bacterial treated midgut, and a rod-shaped biofilm kind of slide was been observed onto the midgut. E and F basically show the Bacillus thuringiensis treated midgut, which clearly shows slight deposition of bacterial rod-shape onto over it. A pesticide treated larval midgut were also been performed like with formed, and iron deposition on slide surface was been disrupted and found. Thus we have extracted the samples from the larvae, and we have confirmed for whether the pesticide derivatives are been performed through GCMS and HPLC, GCMS and other high throughput techniques. So once we confirm that yes, the bacteria in the lab scale is somewhere uh, having an impact to stop the growth of these larvae, our next plan was, can we go for a plant larvae combined study? So we have come up with different setups where we have seed coated the mung plant. Somewhere we have seed coated the mung plant. Somewhere we have sprayed our bacterial culture onto the plants. Somewhere we have inserted the bacteria, our bacteria, uh, uh, into the larvae through Prolex. We have also used the similar treatments for Bacillus thuringiensis via spray, seed coating, as well as insertion through Prolex. and one control was been maintained. Once the plant was been grown grown three till 3 to 4 branching stage, we have exposed all the larvae and we have covered the plant and we made sure that they should get the aeration. So we have plugged the polyethylene bags with so that the air should pass. And at each interval, we have checked the study, and it came into the story very clearly. Into We can clearly see into the right side, right side figure, that B, C, and D, that is Bacillus zeaminensis treated seeds, Bacillus zeaminensis sprayed plant, and Bacillus zeaminensis inserted larval prole have shown the positive correlation where seed coated plants were being consumed by the larvae and they were found to be dead. The consumption of the plant was found to be not much. When it comes to bacterial sprayed, the spray of the bacteria on the plant were fully been consumed and the larvae were found to be dead. And interestingly, into the when the bacteria was being inserted through proleg into the larvae themselves were found to be dead and they were not even able to consume the plant totally. Thus, it showed somewhere a correlation that the bacteria, apart from working as a biofertilizer, can also have a triad to work as a biopesticide. Further confirmation was performed using scanning electron microscopy, which clearly showed the disruption into the larval gut, and deposition of the bacteria was also been found, observed. So, further to confirm what exactly are the proteins present into our bacteria that are somewhere scavenging and targeting the toxins present into the larvae, and thus it is stopping the its infection is under process. So we are on the way to work on the proteins that are present into the bacteria in our bacteria, that will be somewhere helpful for us. So based on that, the summary of objective last objective was that the idea into the mind that whether our bacteria, apart from the detoxification of metals, can also be worked as a pest control agent have also shown a confirmation. The different experimentation that we have performed throughout the study have shown that yes, the efficiency of the bacteria was almost till 48 to 72 hours to target the larvae, go inside, uh, proliferate the proteins that are helping and targeting the toxins and nullifying its activity, which is somewhere helping the plants to grow. And it also confirmed that the mortality rate was found to be increased with reduction with less time, and the efficiency of less infections into the bacteria was also been found. Similarly, the bacteria, which my targeted bacteria, was also been checked for antifungal, antibacterial, and all the antimicrobial tests for all the moon plant specific pathogens, and it has the inhibition rate, higher inhibition rate. Does it confirm that further, after confirmation of the proteins present into the larvae, into the bacteria that is targeting this larvae, will give an impact to the study? So overall conclusion of the whole PhD study, if we focus or come up is that heavy metal contaminated soil specifically is affecting the plants, earthworms. Thus, it is somewhere translocated via roots into the shoots, and it is losing the efficiency of the plants. It is also somewhere harming humans. Similarly, the earthworms which have their uh, habitual place into the soil are been also being affected due to the toxic uh, range of these heavy metals into the plants. And thus, what can be the probable solution? Heavy metal resistant PGP bacteria, when was been exposed to such contaminated and stressful environment, it somewhere have increased the microbial diversity of our specific bacteria into it. Efficient biochemical activities were found to be proliferated, activated. The enzymes got activated, which have helped to scavenge these toxic metals, reduce the toxicity, and biotransformed them into a less toxic form. Thus, it increased the chlorophyll content upon augmentation of bacteria. It increased and maintained the physiology, plant physiology. It also activated it also reduced the oxidative stress by activating the antioxidant enzymes. It also balanced the iron into it. It also have shown the higher uptake activity of these metals into the plants without harming them. And also the biomass of the bacteria was found to be maintained. And as in last aspect was the pest control uh, was also been found to be maintained. So overall, through the study, the two major things what have ended up was, first thing was that the plants whichever been used into the study can be used as a source, because obviously it comes into mind if the deposition of these toxic metals is come into the plant, how the plants can be used. Thus there are certain biological bioleaching or methods through which we can somehow uh, remove or nullify these translocated metals into the form of oxide, and the plants can be used as a into a value added product. Similarly, detection of the protein that have been responsible to fetch the activity and nullify the activity of the toxins that are being produced by the larvae, which is somewhere helping the growth of plant and acting as a pest controller, uh, were the two major findings of the study throughout, which have surely given us an positive impact, that the higher nullification or detoxification of the environment can be achieved through a combination of the living biosystems in combination with bacteria, and can somewhere be a helpful for the research society. So these are the five research articles that have been published from my PhD study, and two have been accepted just last week, which have also a hypothetical oxidative mechanism based on the stress condition. So thank you for giving me and Dr. Malini, Dr. Deshmukh, and all the people who were there here into the presentation, for giving me an opportunity to share in short what exactly my work was, how I have performed the studies, and what exactly was the outcomes of the study, and how it can be somewhere helpful for the research community. Thank you.
Thank you very much, sir. Uh, we are really very much grateful to you for sharing all your resources that you have gathered so far, and like it has been a very vibrant one, and like multi-dimensional, that I should say. So hopefully this will benefit quite a lot number of students to find out new directions for their coming days. So I have one real quick question for you. Can I ask?
Yeah, sure, ma'am.
The point is like you have shown a sort of a niche in the environment where multiple things are involved for this bioremediation. My point is like once the metals get accumulated, etc., etc., but then that is right in that it remains in the locality. Maybe it is they are being drifted away from the toxic source, or from the effluents or whatever the industrial discharges, etc. But then, like, how are exactly is there any possibility that we really get out of, because metals, I like they are not like sort of degradable things like that, right?
Yes. Yes. So, as you have said that metal somewhere can't be degraded, but the mechanism explains that metals can be biotransformed. So, throughout the study, what I have performed, I have shared you that the bacteria what was been isolated was able to tolerate and was a resistant bacteria for the targeted metals. So, what exactly the mechanism, these bacterial mechanism works as, they biotransform the toxic metals into less toxic or non-toxic form. So the accumulated metals into the plants from the soil will be either into less toxic or non-toxic form. Okay. Somewhere many studies were been reported that they are again using the same plant and putting it somewhere into the soil. So what exactly is happening? The plant, the what the metals that were into the plant, they are again going into the soil, but uh, they are into a less toxic or not non-toxic form. That is what the studies have reported. But for your question, the main thing is that these metals can't be degraded for sure, but somewhere we can try to biotransform them using different aspects, so that somewhere a full-fledged detoxification is not possible, but somewhere we can at least initiate to start it using certain probes that they can get non like they can transform. Secondly, as we are talking about that how it is possible, so till we all know that the major source is industries, so the dumping or the maintenance of the toxic waste or any of the excreta that is coming from these industries, the maintenance practices are the major thing that industry should look forward, because of course after once getting a desired product, the waste has been directly exposed into the water and soil. So somewhere it will come, and somewhere there is no stoppage for these toxic contaminants to come into the environment or ecosystem. Thus here the major thing can be somewhere the disposal practices by the industries should be taken into consideration. Then only somewhere the toxicity or the detoxification will be possible, what I feel.
Yes, yes. I was also going to say that only at least the contaminated sites and like what effluent treatment plant, as the name goes by, but how much it works and what is the outcome or benefit, that is a question. Maybe not for all industries, but for quite a number of industries.
Yeah, exactly.
So yes, that's a. So may I please ask you to share your email ID in the chat box so that like students or like
Yeah, yeah,
interested people can get in contact
yeah,
with
yeah,
you.
yeah, yeah,
Yeah,
yeah,
yeah,
sure, sure.
yeah.
I'll just share it. And if any of the doubts or any kind of clarifications
Yes,
or
yeah,
any
others, if
questions, yeah,
anybody else have questions, are you are free to ask, please. Yeah. Are you sharing, please, your email ID? I haven't seen it. Oh, yes. Okay. 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 So thank you all, and hope to remain in touch with you in the future.
sure, sure. Thank you, ma'am. Ha, Ketan?
Okay. So I'm ending the meeting. And so we meet once again in the next week.